This video is about meiosis. The goal of meiosis is to make gametes, sperm and egg cells, sometimes called sex cells. There are two rounds of division in meiosis, meiosis 1 and meiosis 2. The goals are to reduce chromosome number by 1/2 and to increase genetic variation. Just like in mitosis, meiosis starts with a round of interphase. We start with one diploid cell with unreplicated chromosomes. During interphase, the cell grows, the DNA is replicated, and we end up with replicated chromosomes. And then, of course, the DNA is proof-fed. So at the beginning of meiosis, we have one diploid cell with replicated chromosomes. During meiosis 1, this cell splits its chromosomes in half so that two daughter cells are created, but they have half the number of chromosomes as the original cell. You see 2 chromosomes here, and each of these cells only has 1 chromosome. So we end meiosis 1 with 2 haploid cells. So in meiosis 1, the homologous chromosomes separate. In meiosis 2, the sister chromatids separate, and so you end up with 4 haploid cells with unduplicated chromosomes. And we call diploid cells 2N and haploid cells end. These cells that are created are then later used for sexual reproduction. So meiosis is not sexual reproduction. Meiosis creates the cells used for sexual reproduction. In this video, we'll briefly go over the steps that occur during meiosis. So here we're talking about the process of meiosis. Meiosis starts with interphase, like I said earlier. In interphase, the cell grows and DNA replicates. And just like you see in mitosis, the DNA here is in chromatin form. Then we enter meiosis 1. In meiosis 1, you have prophase 1, metaphase 1, anaphase 1, telophase 1. So we'll start with meiosis 1 here. One of the most important parts of meiosis 1 occurs, many of the most important parts of meiosis 1 occur during prophase 1. So this right here is prophase 1. During prophase 1, okay, the homologous chromosomes pair up. And when these homologous chromosomes pair up, we have our first way to increase genetic variation. And I'm going to put all of our increasing genetic variation in orange. This is called crossing over. And you can see this. These 2 homologous chromosomes are lined up, and at these little circle spots called chiasmata, they can cross over and exchange parts of their gene. Okay. But in prophase 1, we still have a diploid germ cell, which is the original cell we start with, right? It's 2N. Okay. And later on, we'll talk about what actually occurs during crossing over or the exchange of genes. Right now, we're just going over the steps. Then we have metaphase 1 here. And again, in metaphase 1, the chromosomes line up at the equatorial plate in the center of the cell. However, you'll notice the homologous chromosomes line up next to each other instead of in a straight line. So the homologous chromosomes line up. And because of that, okay, we have another potential for increasing genetic variation because these homologous chromosomes can flip which side they're on, which flips the side they separate to, right? And so the way that homologous chromosomes line up influences the way they separate, and this is another way to increase genetic variation called independent assortment. Then next we have anaphase 1, which is when the homologous chromosomes separate from each other. They pull apart from each other. And then we have to go all the way down here. Right, this is telophase 1. In telophase 1, we have 2 haploid nuclei. And you can see that, right? We started off with 4 chromosomes here, and now each nuclei, well, actually it looks like, oh no, I'm sorry. We started off with 6 chromosomes, 1, 2, 3, 4, 5, 6. And now each nuclei only has 3 chromosomes. See, you see 3 chromosomes in this nuclei and 3 in this. So now we have 2 haploid cells because they have half the number of chromosomes. That's the end of meiosis 1. And then we get to meiosis 2, which I'll do in a different color. In meiosis 2, we have prophase 2, where you can see we start with 2 haploid cells, right? And you can see their nuclei, they have half the number of chromosomes as the original cell. The nuclei starts to break up, right? And then metaphase 2 is these 2 cells right here. And you can see the chromosomes line up in the middle of the cell. This looks a lot like mitosis. In anaphase 2, in this part, the sister chromatids separate. In these 2 cells. And then you have telophase 2 / cytokinesis, right? And this is the end, and you end up, your end result is 4 haploid cells. So let's briefly go over how crossing over and independent assortment are going to increase genetic variation down here. Like I said earlier, I did all of my increasing genetic variation stuff in orange. So crossing over occurs during prophase 1 of meiosis. And in this process, you have an exchange of genes. And this allows you to mix up the DNA between the different chromosomes, right? So you can see it's this homologous pair. So when they join up like this, they're called a tetrad. And a tetrad is 4 chromatids and a homologous pair of chromosomes. And they undergo synapsis here, which is when the 2 chromatids cross over to each other and genes can be exchanged. And you can actually see this, right? Like if this chromosome originally had genes big A, big B, and big C on its 2 chromatids, and this chromatid over here, let me lighten this up a little bit, originally had genes little a, little b, little c, those genes can be exchanged. Right? And so like we have big A, big B, and C, little a, little b, and c, and you can actually see this exchange down here, right? This chromosome that previously had all dominant genes on both chromatids now has dominant genes on one chromatid on this side, and 2 dominant genes and 1 recessive gene from that other chromosome on this side. This chromosome that used to have all recessive genes has 2 recessive genes and 1 dominant gene. So they traded places, right? So this allows you to increase genetic variation of your chromatids when they separate, okay? Another way we can increase genetic variation is by independent assortment. Independent assortment is how the chromosomes separate, but the thing that allows independent assortment is how they line up in metaphase of meiosis. So in independent assortment, this occurs in metaphase 1 and metaphase 2. The way the homologous chromosomes line up is random. So upon separation, you get diversity or genetic variation here, right? So let's say this was an orange chromosome. This was light blue. Let's see if we can zoom in. And then this was light green. And this was purple. So we're just like, I'm showing you the different chromosomes, okay? The way they lined up in metaphase 1 influences how they separate at the end of anaphase 1, right? So because they're lined up like this, the orange and the green chromosomes will go to one cell, and the blue and the purple chromosomes will go to another cell. So both cells have one big and one small chromosome. But what if the orange and the blue lined up in opposite fashions, right? It doesn't matter as long as the cell has one of each type of chromosome. But now in this example, if they line up this way in metaphase 1, then when they separate in anaphase 1, this cell will have the blue and the green chromosome, and this cell will have the orange and the purple chromosome. So the way they line up in metaphase 1 and then eventually in metaphase 2 influences how they separate in anaphase 1 and then eventually in anaphase 2, allowing you to have a diversity of chromosomes in the different cells, which increases genetic variation. So both of these two, I'm going to zoom out for a second, both of these two, crossing over and independent assortment, occur during meiosis. Okay? But then after meiosis, a third way to influence genetic variation, or to increase genetic variation, is random fertilization. This is which sperm fertilizes which egg in sexual reproduction. There are a ton of sperm and a ton of eggs that are made through oogenesis and spermatogenesis, and depending on which sperm fertilizes which egg, you'll have more genetic variation occurring. Okay, that's it for this video. Thanks for watching.